Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health.
I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Sara Hägg. We have a very interesting conversation for you. We're going to actually talk about sex differences in aging. So what happens and what is different along a male versus female's path as they become older. We'll talk about how Sara actually got involved in studying aging and jump right into some of the big ways that men and women age differently. We'll actually even dive into hormones. So how do estrogen and testosterone specifically influence how men and women age? We'll talk about what happens on a cellular level, a genetic level, an epigenetic level, and even dive into metabolomics and proteomics and the differences with each sex there. The one question I've always had when it comes to aging and women versus men is that women generally live longer than men. And why is that? So a good chunk of our conversation actually dives into this and what we know so far to be true, biologically speaking. We'll also talk about age-related diseases and the ones that men are prone to versus women, and different steps we can take regarding precision medicine that can help us approach aging uniquely for men and women. There are also some suggestions that Sara makes regarding these unique biological factors for interventional treatments. We end with the one misconception people tend to have about how men and women age differently and the surprising things that she's learned in her work about the differences in aging between men and women. A little bit about my guest today. Sara Hägg is an associate professor at Karolinska Institutet in Sweden and leads a research group that focuses on genetic and molecular epidemiological studies of aging. She has a special interest in markers of biological aging and longitudinal associations with age-related traits and drug treatments, a topic where she has published widely. I hope you all enjoyed this amazing episode. And now for my guest, Sara Hägg. Welcome to the Everything Epigenetics podcast, Sara. I'm super excited to have you today. Thanks for joining.
Thank you.
Yeah, I just gave our listeners a brief introduction to you. I know you're leading a research group. You're in Sweden. You're focusing on genetic and molecular epidemiological studies of aging in particular. So this talk we're going to have today is going to be mostly fixed on aging and really understanding the difference between how men and women age. But I want to hear it a little bit from you first. How did you get involved in studying aging?
Right. So I guess it's always been an interest for me. And then I came in to do my PhD in more cardiovascular disease. And after that, I went on to do more work also in dementia-related research and starting to do more genetic and biomarkers. And somehow it all came together on the topic of aging because it was all related to aging, all the things I was doing. So it was a natural continuation to just have them focus on biological aging then as well.
Yeah. Was there any reason in particular you were drawn to cardiovascular disease and dementia? When I think of both of those, I have, I would say, a personal connection with both of them regarding historical events in my family and lifetime. But was there a reason that you chose those two?
Not really. But looking in retrospect, I can now see that actually in high school I did this work already then on Alzheimer's disease. So I must have had an interest from the beginning that I kind of forgot. But then doing the research, I got into this field and it just felt natural because also I have my training is in molecular biology and computer science. So that kind of also has always driven my interest in biology and knowing biological mechanisms. So then studying diseases of aging and having an interest in biology and genetics, and it was a natural way to continue my research into biology of aging, I would say.
Sure. And you have all of these age-related diseases, right? So as you're studying one from or you're going from one to the other, you're probably noticing these same trends and themes. So you kind of took it over under that broader just aging research in general and looking at different biomarkers of aging. So it's super interesting. I want to break it down a little bit further because, as I mentioned, studying aging can be just very broad. So
Yeah.
how do we actually start to dive into some of this science? And I know you're interested in how men and women age,
Mhm.
particularly. Could you give us a little bit more information and set the stage on how we age differently? And then secondarily to that, really, are there any things that are unique to each that stand out to you? I'm sure there are as you'll go through this in detail.
Absolutely. So, I mean, um, for most of the studies that we've been conducting and then doing epidemiological studies on large cohort data, we have a lot of large cohorts in Sweden, so really nice materials to work on. It turned out that more and more of the analysis we did actually had sex specific effect. So whenever we do something now, we always present data also separately on men and women. And I think that should be done basically in all research where it is possible, even in animal work. So it kind of started this interest. And then together with a colleague, I wrote review article then on sex-specific effects in biological aging. Uh, and I would say if you look at any type of biomarker, if you look at the hallmarks of aging in the way that you can quantify that in humans, and so you have telomeres, you look at epigenetic differences, the histone modifications, etc., mitochondrial dysfunction. It turns out that the more you look into it, it seems to be more clear that there are sex differences in all of these different mechanisms that you can measure. Um, and then thinking more and more about this, it's also that whenever you look at the biological data sets, the first three components, principal components that comes out in variability of a biological variability in a data set, it's always the sex-specific effects, it's the aging effect, and then perhaps genetic background. So it's really one of these fundamental differences that you can see in nature. So I think it's a very and of course, the interaction with aging is then very appealing to me in this research field.
Sure. Sure. You mentioned you're working with a lot of large cohorts in Sweden and really in all of the studies. Would it be accurate to say in all of the studies you now do, you're looking at sex specific effects, or you believe they should at least separate the sexes to look at them completely differently because we're going to find other insights maybe dependent on that? Is that correct and accurate to say?
Yeah, absolutely,
Perfect. Yeah. And
absolutely. We always present the main effect with the whole cohort, but in everything we do, we also have the sex-specific effects separately presented.
yeah. Is there any reason that you could think of whatsoever that you would just look at everything together? Like, is there any exception that you could think of? Or you're just saying right now, hey, to any researchers listening here, this is how we should proceed with science in the aging field, or this is my specific request. Because, yeah, you think, I'm not saying it would be easy, but you have all of the outcomes from a specific research study. Let's just start to segregate it and break it down a little bit further.
Right. I mean, of course, that if you pull everything together, you will see the broad picture, you will see the main effect. And that's always what you present first because it's also a power issue that you want to have as many samples together, pulled together as possible. But that said, I think it doesn't cost much to do the separate analysis in men and women once you have that pool data together. So I think it should always be presented, and also for the reason that later on, even if we have power problems in smaller cohorts, when you start to separate in men and women and maybe different age groups, etc., you can always think about the future scenario where you want to do meta-analysis of specific research questions across studies. And then, of course, you have to have the separate estimates as well. So I think it's also important in future perspective that we present these data. I mean, they could be in a supplementary table. Doesn't necessarily have to be the main, but it's so easy to do it. I think for animal research, it's much more complicated, obviously. But even there, once people start to do it, they usually find differences in female and male mice, for example.
Yeah, that would make a lot of sense too, especially with your work as you're going back and you're wanting to do really large review papers, right? Or trying to really gather as many data points as possible. Just having that as a baseline, I think would help expand your work even further. So
Yeah.
that's definitely understood. And, you know, excuse me. We hear a lot about hormones, of course, in aging. I just saw, looking at my Google alerts, I get, emails with epigenetics, epigenetic, aging biology, biomarkers of aging, and all of the above, that there are, all of these hormone clocks being created as well. So that's recently become more prevalent. But can you talk a little bit about how estrogen and testosterone specifically influence how men and women age? What have you found there?
Yeah, so of course, this is an area where there's a big difference in men and women. And this is, of course, the whole starting already in utero when there is, testosterone driving a lot of different mechanisms in men, etc. But for women, up until menopause, the estrogens are really important because they are protective in many ways for cardiometabolic outcomes and many other outcomes related to aging. So it's a really strong link with aging. So as long as you have high estrogen levels, you are protected from many diseases, and it also helps you in many ways. I think interactions with a lot of functions related to aging, like both cellular maintenance systems and also functional systems. And once you reach menopause and these estrogen levels and other hormones start to decline, there is a big shift in aging. So for women, this is really like this dramatic moment, or maybe it's not just one moment. It can actually be anything from 1 year to 10 years or whatever. This transition period, but there is a remarkable difference before and after, and also related to aging. So after that period, once you reach menopause, you actually many women tend to have more similar profiles in terms of risk for different diseases as men. So it's like they are protected, and then once they get to this level, they start to decline much faster in many of the functions of the cellular mechanisms that are we want to maintain as long as possible. But after menopause, then these mechanisms are more similar to what we see in men. Whereas in men, then we have the uh, andropause, which is a similar event, but it's not this clear transition as it is in women, because basically testosterone levels, I think on average, they decline from adulthood. They decline by 1% yearly. So it's a continuous phase. So it's not really clear when men enter andropause, and also, I mean, they still can reproduce because they still have their sperm production, even if they have low testosterone levels, etc. So it's not really comparable. But some men also experience problems with andropause and could have similar manifestations of symptoms and clinical signs as women have, although it's not as common, but it do exist. So absolutely, there is some similarities there as well.
Oh, there's so much information to dive into there. It's like you open this black box, right? A kind of my follow-up question for you, Sarah, after that is, would you be, I guess, an advocate then, or even recommend trying to delay menopause when it comes to women with estrogen supplementation or what have you? I know there's a lot of connections there with, going through menopause and seeing a lot of acceleration within these biological age biomarkers.
Yeah.
And I guess even the andropause, again, that's something I'm less familiar with. You don't hear that talked about as often, but maybe any type of therapies or interventions that we can do to help slow that down as well.
Mm. So I mean, I know now it's getting popular for men in this middle age range to also start to take testosterone levels. I think that is less known. Um, but the research on hormone replacement therapy in estrogen levels for women, that's at least uh, a bit more evidence on that type of and also studies on that. So I think that yes, it may be possible, and I think this is a really interesting future research field as well. We don't have enough information. We need to know more. Is it possible to use these HRT replacements and actually prolong this functional phase in women predominantly then? so that we would maintain function longer and also prolong a healthy life with that. So what is known, I think there seems to be a little bit of evidence, is that if you do it, you need to start early, or you need to start at the right time. You cannot start too late, because if you're already on your transition down, declining in these hormonal levels, maybe there has also already been transition in a lot of the mechanisms. So then it's not beneficial to the same degree anymore. So you have to start early when you still are basically having maintained cellular function. But when you already, I think the timing is very difficult, because you will not start too early when you still have your all your estrogen levels as normal, but not too late either, because then you can see that you don't get the same benefit. Uh, and I also think that it's very interesting talking to a lot of clinicians in this area, where they say that, well, we have these women, they come in and they get this treatment, and they've had it for 10, 20 years, and they don't have gray hair, they look all living and blooming, and it's just amazing. Why don't everyone take this? And I guess the answer is, I don't know. We don't know. We don't have enough evidence yet. So there is definitely more studies needed here.
Yeah, definitely. And that's who I work with, day in and day out, is a lot of these healthcare providers in the cash pay medicine space who are treating healthy people. They're more of your anti-aging, functional, interventional providers. And I would say a lot of them are using the hormone replacement therapy on their patients as well. So it's been really interesting to see that change starting to happen. Even, as my parents get older, my mother and my stepfather wanting to become healthier. And again, their hormone levels are probably declining, but it's something you just don't see. You don't see a lot of attention given to that area when they're just going to their general practitioner offices as well. And Sarah, if you could just on average give like an age for women, maybe men too, when do we really start to see those estrogen levels start to decline pretty rapidly? Is there like a window? I know it's very dependent on the person, but is
Yeah.
there a particular window you could give by chance?
I think the mean age of menopause in women is around 55 years. Um, at least maybe that's mostly based on European ancestry populations as well. But that's the number I have in my head. But, it can, as you say, it can vary very much. It can be, starting already in the 40s. And, some people, it doesn't happen until they're 60. So it's really, really variable, of course. And also the period that you experience all these symptoms and problems is also very.
Right. And you're talking about estrogen supplementation sometime years before that, right? To really start to optimize your levels before going through menopause as
I
well.
guess so. But the problem is
Mhm.
that it's not really tested. Even if you go to your GP, it's like they ask for your symptoms, and then, oh, but you're fine. It's no problem. And it's like, I I think that we also have this misconception that we used to believe that having these type of hormone replacement therapies would lead to increased cardiovascular mortality because of some of the earlier studies. And now we have better treatment. We've seen that that's not really the case anymore. And so it's also a change in mind in terms of the evidence that we have, I think.
Yeah. Yeah, definitely. It's super interesting to talk about and to ponder over. I think hopefully within, my lifetime, I'd love to see some type of biomarker test for menopause, right? Or perimenopause, to be able to tell a woman, hey, you should prepare for this, or, intervene this way. This is how long it's going to last. This is probably what you would expect to feel, just so we can start to normalize that a little bit. So I think there are some research groups working on that, but I'd absolutely love to see that.
Yeah, I agree.
One question this has actually come up a lot, and I don't think I've ever had an answer for this. I think you, out of all people, I'm going to put the pressure on you, Sarah, might be able to answer this with your expertise and your background. We know that just women generally live longer than men, right? They live these longer, healthier lives. Men have usually accelerated biological aging. Um, with almost any biological age biomarker you use. Do we know why that is? I really have tried to do some searching and some digging, and I haven't found an answer, but I'd love to hear from you. Obviously, everything we were talking about, the hormones probably play a role, but what do you think?
Yeah. It's a very interesting question. And I think, um, you can view it from different perspectives. To start with, we have them. the sex-specific paradox, so that we have certain areas like the biomarkers of aging, the cellular markers, where women are better off. So we can see then that women have better cellular maintenance system. So then, of course, people say, oh, it's the telomeres, or it's the epigenetics, or it's all of this, and this explains why women live longer. I don't think that it's just one explanation. I think it's much more complex than that. I mean, we also have the different sex chromosomes and regulation of these genes, etc. But if we would take one step up and look at it from a theoretical point of view, I mean, evolution, in some way, men are more susceptible to be fit early in life because they need to, be the predominant male that can reproduce and produce offspring. Whereas women are a little bit, more careful because we need to be the one that provides for the offspring for a longer period of time. So just thinking about the biological human beings that we are, it's really a difference in the way that nature tried to modify this. And one example of this is that if we look at autosomes, there is not so many differences in terms of genes for men and women. But there are some differences in anthropometrics. So we can see that there are sex-specific effects, the way that we have genetic variants coding for height, for example. So how tall we get, which is one of these things that men, they like to be tall because, the taller they are, the more fit, the more, easier it will be to find someone to produce an offspring. Whereas women, we don't want too tall individuals because if we have a very tall mate, we may get offspring that are very big, and then we can't give birth to them. That's a risk. So women try to put the balance on that. So that's actually where we see a sexual dimorphism effect on these genes. So I think that's really interesting. So it also speaks to this fact that there is a way that nature is trying to balancing this fitness versus survival effect, where we have different pressures on us as men and women. So in that way, I think it's natural to think that women have better maintained cellular maintenance system because that's what we need to survive longer, to be able to repair and restore and keep our cellular functions intact. Whereas men, they put a little bit more risk into being fit early in life, with a cost that later in life they don't have the same possibility for repair and in these cellular functions. So I think that there is a fundamental difference here that is somehow then going through all of these complex mechanisms that happens in life, and the differences we see in men and women is somewhat perhaps a result of this difference.
That's interesting. I'll tell you, that's the best answer that I've gotten. So I'll take it. No, I think it makes a lot of sense. It's like you mentioned, it's this underlying evolutionary effect almost, right? As time has gone on. And my mind actually goes to more of the future, if we'll ever see this change. Maybe not men living longer than women, I would say. I never want to say never, but it may be leveling out a little bit more, or and the reason I say that is because now we're starting to see a lot more women, go through the IVF process, right? Even on their own, or there's just a lot of changes, I think, happening in the world with just overall reproductive health. So I'm wondering if we'll start to see kind of evolutionary changes even down to the cellular level as well. It'll be interesting to follow that.
Yeah, I think that would be interesting. We don't know how that will affect. But I also think that one thing that is in support of this is that you can see in societies where, the gap between men and women in terms of, income or in education, all of these social factors, where the gap is getting smaller, then also the survival difference is getting smaller. So it speaks to the fact that there is some sort of social context there that could perhaps help to close the survival gap. But yeah, I don't know how all of these new treatments and the IVF and all of those things will actually influence and change our evolution in some way.
Yeah, yeah. I didn't even think about the social aspect of it all, but it's all interplaying together.
Yeah.
You brought this up a couple times, Sarah, actually. I'm curious about just the role of genetics here too, right? We talked about having different chromosomes, or sex chromosomes. How much does our DNA, if you want to look at that almost as like an aging biomarker of some sort, influence the differences between men and women's aging?
Um, well, most of that is in the sex chromosomes, I would say, because um, I mean, overall in aging, our genes are uh, surprisingly have a quite low effect on the overall survival in life. So, I mean, the environmental contribution is much bigger, at least if we speak about general populations. So, also in the sex specific effects, I mean, we haven't seen much sex difference in genetic variants in all the other autosomes, all the other chromosomes beyond the sex chromosome. So the the one that I brought up in the difference in height, genetic variants, that's one example, but there aren't actually that many
So
here.
very few. Yeah, it's going to be more about the epigenetic interactions,
I
right?
think so. It must be about regulation and how the hormones interact with the regulation processes in the bodies, for example, because we're more or less born with the same genes, basically.
Mhm. Yeah, exactly right. And then, so we talked a little bit about genetics. You gave that really good example of the height differences. What about just epigenetics in general? I know that's a really, really broad question. I can't not bring it up with my podcast being named Everything Epigenetics. So you
Yeah.
have your lifestyle choices. I mean, your diet, your exercise, your sleep, your stress, all of those. Um, are I mean, it's this is a hard question to ask almost because it's very dependent just on the individual person, regardless of whether you're a male versus female. But are there any lines that can be drawn that say, epigenetically we're seeing maybe something a little bit more favorable or less favorable with male versus female?
Yeah, I mean, you're absolutely right that there are so many things that influence, but on on all levels, when we look at DNA methylation or histones or whatever, we see that there are differences in men and women, and we have done a lot of studies then, of course, with the epigenetic clocks, and if you also look at those levels when you age, you see the sex difference again here that that women tend to have overall a better maintained epigenetic clock compared to men. And that we see that basically across all different types of epigenetic clocks. So there is absolutely this differences on the epigenome. And I would say it's you would probably find it regardless of what you're looking at. That's my guess. But as you say, of course, then it's the next level that, in biological variation depends on so many things. So, of course, the individual variability is still larger, I would say. So, I mean, we speak in general terms, but of course, there could be one woman that looks completely different. And then you compare to that man that looks more like this group of women or whatever. I mean, you can always find exceptions to that.
Sure. Sure. No, that's good to note. And we've talked a little bit about genetics and epigenetics. You talked about different epigenetic modifications, DNA methylation, histone modification, etc. And telomere length as well. There may have been a couple others that you mentioned. But in terms of the studies that you're doing, what other biomarkers are showing us these aging differences between men and women? And can you just expand on that a little bit? Like, how much can we actually see from those markers? Even what's your favorite to look at, right? Like when you perform a study, are you including all of these biomarkers? I know cost can be a really large factor as well, but even if you want to walk us through one of your studies and some of the interesting findings there.
Right. So I think one of the new things we're doing now is proteomics, and also metabolomics has been ongoing for some time, and doing all of these specific clocks. And here we can see that we have one project now that we're doing. Of course, like a lot of other groups are doing this organ specific clocks, but we're doing it on Swedish samples. And there we can see that, of course, there are proteins that are sex specific, and you can also then develop organ specific clocks for men and women separately, and you will have a little bit different results. So I would say that you can see this on all of these molecular markers, biomarkers. But another type of marker that we're looking at is functional markers. So we, in the cohorts we try to assess as much as possible. So we also look at, muscle strength in different ways. So we have lung capacity, so you get to the aerobic capacity a little bit, and also, grip strength and walking speed and things like that. And here, of course, we know that men perform better than women because, they are bigger, taller, they have larger hearts, and bigger volumes in their lungs, etc. So they are overall stronger and perform better in these markers. Um, because they put their energy on the fitness, so it makes sense that's what we see. Um, but yeah, so I think that's also another area where it's interesting. And then of course we can again see the sex paradox, because if women are better off in the cellular level, then when we move to the functional levels, we see much better results from men compared to women here. Yeah.
Yeah, exactly. No, that's super interesting. Yeah, in the space that I am in, obviously my company, True Diagnostic, we offer the Symphony Age commercially, created from Dr. Morgan Levine and Albert Higginshen and Raghav Sehgal, and that's the aging of the 11 different organ systems. But they're not separated based on sex. And I want to go back now and separate all of them and rerun them because, like you mentioned, just that functional measurement of, like, we have the heart age, the musculoskeletal age, the metabolic age, right? They're
Mm.
gonna have you're gonna have different trends in men versus women.
Mm.
Again, we run the algorithms on each of the individuals, and then we'll compare them to the population based off of, female versus male, but you're not running the clock. The clock wasn't created just on men or just on females. So you have my wheels turning and spinning on how to make these predictors even better. Mhm.
Yeah. So I mean, it's always, you have to make these choices all the time. Like, should you prioritize a bigger sample, or should you get to this specific sex effect, or it's like all of these choices. So of course, in different ways you can do it.
Exactly. It's hard too. There's a hormone age on there that, unfortunately, they were only able to use IGF-1 and DHEA for the creation of the algorithm, because that's all that the biobanks have, right? And it doesn't make it any less predictive of the diseases because they validated it in a really nice way. But we also understand the limitations of it, where it's not grabbing estrogen, testosterone, progesterone's other hormone markers too. So hopefully we'll get some more biomarker data and be able to make that a better predictor. Another question for you. So you have these biological, you have these biomarkers of aging per se, right? What about then, coming back to the beginning of our conversation, these specific age-related diseases, right? We know that there are certain ones that men are more prone to and women are prone to. So what might that tell us about how each gender ages? Do you look at that in some of your studies, and then can we intervene based on that?
Yeah, yes, we do. We try to. I mean, so there are different things. There is this, just looking at statistics, how many men and women have this type of disease, etc. And then, of course, you can also look at, okay, so but what's the risk of getting that disease, given that you're a man or a woman, depending on if you look at a biomarker, for example, as an exposure. So there are different ways of looking at it, but um, what I can say is that we know that there are differences. So men usually have more of the cancer diseases. I mean, cancer is, of course, a broad spectrum of different cancers, and reproductive cancers, etc. That they're different in different sexes, obviously. But then women have more of the women tend to respond better to, to vaccines, and have a higher, immune function in general, in terms of response to pathogens, etc. But that also leads to a bigger risk of having autoimmune diseases. So in women, we can see that that happens at least before menopause. They have higher risk for that. Whereas men then have more of immunosenescence and maybe this aging problem. So there are differences there, and of course we know dementia is much more prevalent in women, and that could be many reasons for that. So absolutely, there are differences here, and we know many of these, the hallmarks of aging, many of the processes here have quite overlapping functions with hallmarks of aging or hallmarks of cardiovascular disease, and there are also hallmarks of neurological disorders now. So all of these things also speak to maybe a little bit of these sex-specific mechanisms as well that's underlying these different diseases.
Yeah. And talking about all of the differences that we do know and that are apparent today, do you think a lot of these, again, the cash pay medicine space, more of these integrative, holistic based clinics will start to take more of an approach? Because we think probably men and women should be thinking about aging differently, right? Regarding their health and vitality. I think we do see a lot of clinics offering, women's health or men's health. But do you see your work really starting to maybe dig into more of that interventional space to be able to say, hey, this is how you're aging. This is how we should, of course, treat each individual, but especially just starting with each sex.
Yeah, absolutely. I completely agree. I mean, starting with the different sexes is like the first thing you can do in precision medicine then. But I mean, for one thing, a lot of women are still not included in clinical trials, so we don't know exactly that they respond the same way to men for different treatments and in different age groups, etc. So that is one thing, and also in general, when we give advice on lifestyle factors and changes on that, there is also a big difference there. So how men and women both in terms of diet, maybe what they would benefit from, and also in physical activity, because they have the difference in functional performance. They also respond differently to different physical activity, strength training, or more in cardiometabolic fitness training. So it's all of these differences we could absolutely do better. And I was just looking at the WHO and their guidelines for dietary intake and for exercise, and there are absolutely no specific guidelines for men and women. It's all just one big thing. And also for older individuals, there may be a little bit differences in some of the guidelines, but not clearly enough. So I think we can do much better here.
Yeah, that really just surprises me. It breaks my heart a little bit, that was kind of my next question as well. Yeah, if there's even those recommendations out there. I mean, that seems like the bare minimum for like our health as a species, as an entire society, is to say we know you two are different. Here's just some little tweaks or minute differences we may want to make in terms of recommendations to have better outcomes and to live longer. So I definitely
Yeah.
hope that changes in the future. And this kind of aligns with just that. What's maybe one misconception or the biggest misconception that people tend to have about how men and women age differently? Does anything stick out to you?
I mean, maybe it's this thing that you often read it still, like, oh, we found this difference in men and women. So it must be the one thing that explains the survival difference. So they always, whenever they find, when it was the telomeres, it's like, oh, this is the whole explanation. But I mean, it still amazes me that people can think that it's really just this one thing. I mean, it's very often with biology, it's so complex. So I think that it's not going to be just one single thing that will solve the whole thing. It's much more complex than that. So
Yeah, so maybe on the surface, of course, there's going to be a sex difference, right? We should expect that, I would say, in every single study, right? If we didn't, then maybe we're doing the study wrong. But obviously, that's again an easy way out. We really have to dive into then each individual sex, I think, a little bit further to start to explain those differences and get closer and closer to the truth. So yeah,
yeah.
that definitely makes sense. And then
Mm.
we're getting to the end here as well, Sara. What's one surprising thing you've learned in your work about the differences in aging between men and women? Or just something that, it could even be what you're most excited about, why you keep coming back to this topic over and over again, and, why you're dedicating your life's work to it.
Right. So one thing that we haven't really touched upon is like the later life when you get multimorbid, you actually have these diseases and symptoms developing, and you get frail. So in the frailty perspective, there are a lot of differences. So it turns out that there are both societal and biological differences in men and women. So men seem to be, women outlive men, but they do it with worse health. So they actually have more multimorbidity and being more frail in the end of life, and for a longer period than men. But they still outlive them. So they do it with this, this sick span that we call it in the end of life. And I think that that's really sad. Like men, they survive being fit all the way to the end, and then once they get sick, they just die basically. I mean, it's an exaggeration, but I wasn't really aware of the end of life, the later part of the life where there is apparent sex differences as well. But maybe we can do better there as well. I don't know.
Yeah. So that's surprising to you. That's something that you're looking into a little bit more as well. Because yeah, then as a woman, it's like, do I really want that? Sure, we live longer, but it's that health span versus lifespan. You said six span type of argument. So it's almost hard to think about.
Yeah.
And then are you doing any interventional trials as well right now? Any interventions with any of the cohorts that you're doing too, looking at extending lifespan?
We don't, but that's obviously something I would like to do, and I've started collaborations with some health tech companies in Sweden. So we're hoping to do that soon, because also part of this is like, the whole problem with the healthcare sector, that they're treating the old and the sick people. But what if we can, put some of that into more preventive healthcare, so we can actually do something earlier in life. So we're hoping to do some studies of that, and we have some plans. So in the coming years, I really hope to do that.
Perfect. Well, I'm so excited to bring your work to light, and I can tell you're just super passionate about it. So I'm excited for what's to come. We've come to the end of this amazing podcast, though. I do ask all my guests one random question at the end. So, Sara, if you could be any animal in the world, what would you be and why?
Oh,
It's a curveball.
yeah, that's a good question. I would probably like to be like some sort of bird where you can fly.
Oh,
I think that would be really cool. But maybe you wouldn't. Well, if you're a bat, then I would live long. So maybe a bat.
there you go. We'll take it. That's a great answer. No, I love that answer. And then for anyone who's listened to this podcast who wants to connect with you, obviously they can Google your name, they can look at your Google Scholar, all of that good stuff. Do you want to point them in the direction of a lab website or anything else?
Yeah, so I'm a bit bad with the lab website, but I do have a good profile page on Karolinska Institutet. So if you go into ki.se and Google my name, you will find all of that information.
Perfect. Well, thanks so much, Sara. And thanks everyone else for joining us at the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn how. Thank you.
Thank you.